Okay, hello everyone. Okay, we will start discussing about the important topics for part 4. Today we will discuss majorly about the DNA repair mechanisms. So friends, these are all the details about the embark batch for the CUCET 2022, which is going to be commenced from the 9th February. And this is the details about the gladiator batch for the GAT-B 2022. So in these two batches, the syllabus for the CUCET and GAT-B can covered from basic to the advanced level. Okay, and friends, this is the detail about the JAM 2022 full length mock test that is going to be happened tomorrow. So in the morning section, physics, biotechnology, statistics, and in the 2 p.m. mathematics as well as the chemistry. Right. So to get a 10% discount, so please kindly use my code Chandu Live. Right. Okay, friends, let's try to understand the DNA repair mechanisms. Okay. So friends, what kind of questions we can expect in the DNA repair mechanism? We can expect, see, it's a very important concept focus. We can expect the question from the type of DNA damages. We can get the question, what kind of repair mechanisms are involved in the repairing of this particular damages. Most importantly friends, the enzymes involved in the repair mechanisms, then friends, followed by the most important mechanism of action. Mechanism of action. Friends, these are the various questions which we can expect in this particular DNA repair mechanism. It's a very important concept friends. You can expect in the 1 marker question also. Even friends, you can expect in the 2 marker session also. Okay, let's try to understand one by one elaboratively. So let's start with the first one, the type of damages. So there are many type of DNA repairs or DNA damages are there. We called deep urination, deamination, oxidation, alkylation, dimer formation in the bases, mutations caused by the base analogs, and mutation caused by the intercalating agents. First friends, we will understand different kinds of damages. Later we start understanding what kind of repair mechanisms are involved in repairing this particular damages. Okay, let's start. So the first one, what is the first one? Depurination. Friends, the name itself indicating that depurination means removing of a purine. I mean to say friends, removal of the base. That's what we can call it as a depurination. Depurination involves the loss of purine bases which are adenine and which are guanine from this particular DNA. Do not forget this point. What friends? Depurination involves loss of purine. See friends, it may be loss of purine also, it may be loss of pyrimidine also. All of you look into the diagram. This is a nucleotide. Is it? What does the nucleotide consist of? Nucleotide consists of sugar, it consists of base, and it consists of phosphate group. We all know very well the sugar and base connected with each other by an important bond. We can call it as what friends? N-glycosidic linkage. We know that very well. Now friends, what is the point I want you people to understand here? Here there is a breakage of the N-glycosidic bond. So you only try to understand friends, whenever there is a breakage to the N-glycosidic linkage, immediately what is happening friends? This particular base guanine get completely released out. Now you only tell me, is there any purine present here? No. So that's what we can call it as what? Depurination. You can call it as a purinic site. And friends, please kindly don't think that only purine can be removed out. No, even pyrimidine also can be removed out. So that's why we can call it a purinic site. Even we can call it a a pyrimidinic site. Simply we call it as a basic site. A basic site means what you have to understand. A means no base is present. That's why friends, we will be calling it as a basic site, a purinic site, a pyrimidinic site. Then friends, followed by the a basic site. Very simple. That is the first damage. We call it as friends depurination. And one more important point you have to remember friends, what kind of repair mechanism is involved in repairing this particular damage. All of you remember friends, we have to use an important repair mechanism, base excision repair mechanism. Please kindly remember this friends. For which damage, which kind of repair mechanism is used, that thing we have to compulsory remember, which we call base excision repair. Okay, what is the next type of a damage? Friends, next type of damage we call it as deamination. Friends, actually this deamination is a very important kind of damage which we have to remember compulsory. Deamination. So please kindly don't confuse. It is not a transamination. It is not transamination. That is different, right? Transamination is different, where there is a transfer of amine group. That is not. Here deamination is what friends? Removal of the amino group. Okay, everyone try to understand this. Deamination. See friends, because of the removal of a particular amino group from the base, it is going to be converted into another. Look into this. So you have to remember this without fail. Because of removal of what base, it is going to be converted into what? Cytosine. Upon removal of the amino group, cytosine can be converted into uracil and 5-methyl cytosine. See why we are calling it as a 5-methyl cytosine? There is a presence of the methyl group. Presence of the methyl group at the 5th carbon. That's why we call it as a 5-methyl cytosine. Upon removal of the amino group, 5-methyl cytosine is going to be converted into what friends? Thymine. Now you look into the adenine. What do you have to understand by the word adenine? Adenine give rise to what friends? Hypoxanthine. Please kindly do not forget this. Next one is what friends? Guanine. Upon removal of the amino group, guanine is basically converted into what friends? Xanthine. Cytosine converted into uracil, 5-methyl cytosine converted into thymine, adenine converted into hypoxanthine, and friends, the guanine is basically going to be converted into what friends? Xanthine. Please kindly do not forget this particular statement. Cytosine to uracil, 5-methyl cytosine to thymine, adenine to hypoxanthine, guanine to the xanthine. You have to remember every single step without a fail. So the next one, oxidation. What is this oxidation? Means treating of the DNA bases with certain oxidizing agents. That is what exactly friends we will be calling it as oxidation. See, there are more than 20 base lesions have been identified. Only a fraction of these have received appreciable study. Most notably what? 8-oxo-deoxoguanosine. Everyone please kindly look into this. This is a guanine. You all commonly know that friends, guanine has to pair it with what? Cytosine with the 3 hydrogen bonds. Correct na? So friends, when the guanine treated with certain agents like H2O2, like OH minus, so during this time what is happening friends? The treating of guanine with certain oxidizing agents, it is going to give rise to what friends? It is going to give rise to the 8-oxoguanine. What friends? It is going to give rise to the 8-oxoguanine. Now guanine originally paired with the cytosine, we know very well. But 8-oxoguanine never ever pair with the cytosine. Do not forget that particular statement. Next, Next, what you have to understand, friends, next one is what we call alkylation, which means treating of the DNA base with the alkylating agents or the methylating agents. Kindly do not forget the statement. Okay, alkylation damage of DNA is one of the major type of insults which cells must repair to remain viable. Please kindly do not forget this particular statement. See friends, all of you try to understand it. Listen to me. When we treat the DNA with a different type of alkylating agent and methylating agents, they completely change the transition. So everyone look into this, friends. Cytosine and guanine. Cytosine, whenever it is treated with an important mutagen or an alkylating agent, what is that? Hydroxylamine. Upon treating of the cytosine with the hydroxylamine, what it is going to give rise to? Cytosine converted into what, friends? Hydroxylaminocytosine. Why? You can see here, to that particular amino group, the hydroxyl group was attached. That's why we are calling hydroxyl aminocytosine. Now that hydroxyl aminocytosine basically associated, see, originally cytosine will associated with the guanine, we all know very well, but that is not happening here. The hydroxyl aminocytosine, instead of associating with the guanine, friends, it is associated with what? Adenine. Then the entire transition will change from CG to TA. It is one of the dangerous mutation, we can say that. Next one you try to understand. Guanine. Guanine treated by an important alkylating agent which we called, friends, MMS. We call it as a methyl methane sulfonate. Guanine, upon treating with this particular methyl methane sulfonate, see, this methyl group come and attaches to which carbon? At the 6th position. Then what do you call it as? We call it as O6 methyl guanine. Now friends, this particular O6 methyl guanine, originally we know, right, guanine has to be paired with the cytosine, but guanine, upon addition of the methyl group, it never associated with the cytosine. Instead of associating with the cytosine, the O6 methyl guanine can be associated with the thymine molecule. Then the transition converted from GC to AT transition. Do not forget the statement, please. Next, another very important kind of damage, that is what do we call dimer formation in the bases. And people will think that only pyrimidines undergo the dimer formation. No. So there is no doubt here pyrimidines are undergoing the dimerization, but along with the pyrimidines, even purines also undergo the dimerization. No doubt about it. But when compared to the purines and pyrimidines, pyrimidines undergo the more more dimer formation when compared to the purines. Best example is what? Pyrimidine. Good morning, Vasudevo. Please continue. Okay. There are two thymine molecules. This is one thymine and this is another thymine. Friends, we all know very well that thymine has to be associated with what? Thymine originally has to be associated with the adenine. You know very well. But here, what is happening? Two thymine molecule associated with each other, and friends, they formed a dimeric bridge. What they formed, friends? They formed a dimeric bridge. So when the thymine exposure to the high intensity of sunlight, there is a formation of a bridge between the two thymines. This is what exactly, friends, we will be called as a pyrimidine dimer. And one more important point you have to remember, since this particular dimer appears a chemical or look like a chemical, what was that? Cyclobutane. That's why, friends, that thymine dimer, we can also call it as what, friends? We can also call it as a cyclobutane photodimer. What do you call it, friends? You will be calling it as a cyclobutane photodimer. Please kindly don't forget this particular statement. What do you call it? A cyclobutane photodimer. Now friends, suppose example, if we don't break this particular thymine, if the same information passes to the next generation, it becomes dangerous, and this is one of, you can remember, pyrimidine dimer or the thymine dimer is one of the major reason for the formation of melanoma. Formation of what, friends? Melanomas, which are a type of skin cancers. Which are what, friends? A type of the skin cancers. Please kindly do not forget this particular statement under any circumstances. Melanomas are one of the major reason for the skin cancers to occur. Don't forget this. Now next one. Mutations, and very important point, friends, how we can repair this particular cyclobutane photodimer or pyrimidine dimers. We will be treated with an important repair mechanism. What do we call it as? A direct repair. And you know, right, the direct repair mechanism also called as what? It is also called as a photoreactivation. Photoreactivation. Any of these two, either direct repair or the photoreactivation, is involved in the breaking down of this bridge and separate the two thymines. Next, mutations caused by the base analogs. What you have to understand by the base analog? Friends, don't get confused with the base analog. See, when I use a word base analog, you people has to remember one of the most important point. Base analogs, friends, actually they are similar. Similar in terms of what? They are similar structurally. They are structurally similar, friends, but they are not functionally similar. That is what base analogs. Let's take an example of some base analogs, such as 5-bromodeoxyuridine or 2-aminopurine. They are the one which is incorporated into the DNA during the process of replication. You know, no, 5-bromouracil is the most common example for the base analog. Friends, if you exactly look into the structure of the 5-bromouracil, everyone please kindly look into the structure of 5-bromouracil. 5-bromouracil, it's behave like a thymine. What, friends? 5-bromouracil, it is just to behave like a thymine, but what is the statement you have to remember? It is not, it is not exactly functioning like a thymine in the rare state. First of all, friends, why I am using a word normal state? Normal state means where it is not undergoing any kind of a modification, either structurally or functionally. There is no chemical modification. That's why we can call it as a normal state. So in the normal state, the 5-bromouracil behave like a thymine. But friends, everyone please understand, in case of a rare state, see, in the normal state it behave like a thymine, so undoubtedly it will go and pair with the adenine, there is no problem. But friends, the same 5-bromouracil behave like what? In the rare state, it is not at all behaving like thymine, it is behaving like a cytosine. Then cytosine paired with what, friends? Guanine. So originally the transition should be thymine to adenine. This transition completely changed to cytosine to guanine. See, which is one of the most important mutation, we can say that. So base analogs are those which are structurally similar, but they are functionally entirely different. Next, another very important one, friends, mutation caused by the intercalating agents. What are intercalating agents? All of you remember, friends, intercalating agents are very small, thin, a plate-like structure. Thin, a plate-like structure which are hydrophobic in nature. What friends? Intercalating agents are hydrophobic heterocyclic ring molecules that resemble the ring structure of the base pairs and include ethidium bromide. Very important point you people has to remember is the examples. Intercalating agents are the hydrophobic heterocyclic ring molecules which resembles the ring structure of base pairs. And what you have to remember friends, the best examples. They can ask you which of the following is an important example for the intercalating agents. Ethidium bromide, we can also call it as ETBR. Then acridin orange, and friends actinomycin D. All of you has to remember, among all the intercalating agents friends, the most important intercalating agent will be the acridin orange. Please kindly do not forget this particular point. What is the most important one? The most important one is always acridin orange. Do not forget. Insertion of these agents distort the DNA double helix, thereby interfering with the DNA replication, transcription and repair. Which means that these intercalating agents damage or break or create a gap in the DNA double helix. Because of that, intercalating agents may interrupt a variety of processes. It may interrupt replication also, transcription also, as well as the repair also. Now you have to understand, please kindly don't think that a particular enzyme will come and insert this particular intercalating agents. No. Intercalating agents on its own, randomly they can insert it. There is no requirement of any enzymatic system to help the intercalating agent to insert in the DNA. No. Intercalating agents on its own it can be introduced. Kindly do not forget that. Mutation, see, you have to remember friends, intercalating agents basically causes what? Intercalating agents basically causes frame shift mutation. What they cause friends? They are basically causing what? Frame shift mutation. Please kindly do not forget this point. What is this frame shift mutation? Either by addition or by the deletion. Especially friends, intercalating agents cause the most dangerous type of the mutations exclusively by the process of deletion only. Please kindly do not forget this particular point. Most of the dangerous mutations are what friends? By the process of the deletion only. Don't forget the statement. Suppose example, you can look into this. The yellow color, thin plate-like structure. Correct na? Thin plate-like structure which is hydrophobic in nature. Here intercalating agents will associated or inserted over there. Now when you remove the intercalating agents, what is the point you have to understand? Whatever the base where the intercalating agent is inserted, even that particular base also completely comes out. So which leads to the deletion. Okay, deletion of the base will change the entire reading frame, which leads to the non-functional protein or the hyperfunctional protein can be synthesized, which is one of the most dangerous mutation. So friends, right now whatever we have studied is what? Different type of damages we studied. What are those? You can observe deep urination, deamination, oxidation, alkylation, dimer formation, mutations caused by base analogs and mutations caused by the intercalating agents. I want every one of you to read and remember every single example of this particular mutations. You have to remember. And most another important point, which repair mechanisms are involved in repairing this. That is most important point you have to remember. Now next, another important point. What are the various types of the DNA repairs? You know very well, mismatch repair, base excision repair. See, always remember the short names. Mismatch repair also called as MMR. Base excision repair also called as BER. Nucleotide excision repair also called as NER. Friends, this direct repair we can also call it as photo reactivation. What do you call, friends? We call it as a photo reactivation. Next one is what? Recombination repair. The next one is what? SOS repair. Friends, SOS repair, what do you call it as? You call it as error prone repair. Please kindly remember all other names. And friends, there is one major difference between direct repair when compared to the all other repairs. What is that major difference? Everyone try to understand. In all other repair mechanism, whichever the, listen my statements, in all other repair mechanism, whichever the part of the DNA that get damaged, we will remove that particular part of the DNA and we will replace with the new one. Once again listen, in case of all other repair mechanism, whatever the part got damaged, we remove that part and we will replace with the new part. But friends, in case of a direct repair, we never ever remove the damaged part. What do you mean by that? So without removing the damaged part, there itself we will repair that particular damage without removing the base or removing the nucleotide. That's why we can call direct repair or photo reactivation, one of the unique DNA repair mechanism we can say. And you also know very well, photo reactivation is the first identified DNA repair mechanism. It is the first identified DNA repair mechanism. Kindly don't forget this. Okay. So friends, whenever you are reading the every single repair mechanism, what exactly you have to remember? First thing you have to remember, major thing friends, you have to remember enzymes involved in every repair mechanism. Enzymes involved in every repair mechanism. Second point friends, you have to remember mechanism of action. I mean to say how every single enzyme is basically acting. You should also remember diseases associated with the failure of the DNA repair mechanisms. So friends, in every single repair mechanism, there are different kinds of enzymes are present. Good morning Neha. There are different kinds of enzymes are present. We have to remember every single enzyme. Okay. Yeah, Neha, please watch the recorded. So in this particular class, I have explained all the type of the DNA repairs involved. DNA damages involved. So in the tomorrow class, we will discuss all the type of repair mechanism, enzyme involved and their mechanism of action. Okay. In this class, I have completely covered the type of damages. Okay. Okay, dear. Huh? Thank you all. I'll see you in the tomorrow class. Okay. Tomorrow also same time, 9 a.m. Okay. Thank you all. Thank you so much.